Hello and welcome to today's webinar. On behalf of European Pharmaceutical Review and Thermo Fisher Scientific, I'd like to thank you all for attending. My name's Hannah Balfour. I'm the Assistant Editor of European Pharmaceutical Review, and I will be your moderator today. Today's speaker will be Shailesh Karavadra, Applications Manager at Thermo Fisher Scientific. Following his presentation, we will move on to a live question and answer session where you can pose questions to Shailesh. Please remember, you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So, without hesitation, I'm going to pass over to today's keynote speaker, Shailesh Karavadra.
Thank you very much for attending this webinar today. My name is Shailesh Karavadra. I am Applications Manager for Thermo Fisher Scientific. I've been working with Thermo Fisher Scientific since 10 years, and previously I have had analytical experience of implementing various vibrational techniques in chemical manufacturing, pharmaceutical analysis, and environmental and food and beverages. Today's topic is to look into how to improve the efficiencies and increase the return of investment by adopting so many of the molecular spectroscopy techniques like Raman, near infrared, infrared in pharmaceutical manufacturing. In today's agenda, I will talk briefly about vibrational spectroscopy. What is vibrational spectroscopy? How infrared, near infrared, and Raman spectroscopy differ? Then moving from there, we are going to see some implementation guidelines which are recommended by USP and EP regarding the strategies to be used for implementing vibrational spectroscopy. There is a separate guideline for validation. We will touch that point too. From then on, I'm going to discuss the most widely done identity testing of raw materials, and we are going to look at a case study of that. There are traditional titration techniques like Fisher, which can be also replaced by near infrared or sometimes Raman. Then there is a case study of replacing gas chromatography for solvent exchange. Hot melt exclusion is widely done for many of the pharmaceutical products, and we have a very good case study which shows the implementation of NIR. Then moving on to various final product analysis, for example, replacing HPLC assays or PDMS quantitative analysis with FTIR, looking at the assay using quantitative analysis for liquid dosage form. And then finally, we are going to look at the final product, especially for API distribution, excipient distribution, or looking at the foreign particulate matter in injectables or parenterals. And lastly, I'll talk about our solutions and service and support. Moving on to vibrational spectroscopy and comparing all three techniques. As we all know, vibrational spectroscopy characterizes the materials or molecules by the frequencies of their molecular vibrations. There are three main techniques: Raman, mid-infrared, near-infrared. Mid-infrared is also sometimes called MIR or even FTIR. A chemical bond in a molecule can vibrate in many ways, and each vibration is called vibrational mode. And there are different kinds of vibrations, stretching and bending. And the two widely used spectroscopic techniques based on vibrational energy are Raman and infrared. And infrared can be divided, into mid-infrared, far infrared, near infrared, and so on. When a sample is irradiated to a beam of incident radiation, it absorbs energy at frequencies characteristic to that frequency of the vibration of chemical bonds present in the molecules. This absorption of energy by vibrating chemical bond results in an infrared spectrum. Raman spectroscopy is based on absorption of photons of a specific frequency, which is followed by a scattering at a higher or a lower frequency. Modification of the scattered photons result from the incident photons either gaining energy or losing energy to the vibrational and rotational motion of the molecule. There are different selection rules for the vibration to be active. For example, a vibration is IR active if there is a change in dipole moment during the vibration. And Raman spectroscopy involves inelastic light scattering, and the selection rules are different from those that of IR. Raman active molecules must undergo a change in polarizability of the molecule. In a nutshell, we can say that infrared and Raman spectra provide complementary information, and between these two techniques, all vibrational transitions can be observed. Looking deeply at a technique overview, so infrared and near infrared uses sources. Infrared typically a glow bar source which emits in mid IR range, and near infrared source is typically a tungsten bulb, very much like our visible light bulb, which emits the near infrared wavelengths. When we look at an absorbance or a reflectance technique, you have a source which emits in multiple wavelengths, you have a sample, and then at the end you have a detector which either looks at the transmitted light or a reflected light, and depending on that you have an absorbance or a reflectance spectrum. However, with a Raman scattering, we use a single wavelength laser source. Typically this are 785 nanometer laser, so you can say it emits in near infrared or far infrared, for example. Most of the time it's 785 nanometer laser, which is typically used in pharmaceutical analysis, but we can do the analysis with 532, 633, and so on. When you irradiate a sample with a single wavelength laser light, multiple discrete wavelengths are emitted. These are collected by a detector like a CCD. And as I said before, the modification of the scattered photons result from the incident photons either gaining energy or from losing energy to the vibrational and rotational mode of the molecule. There are 4 major sampling techniques in FTIR. You have a transmission, a classic method, which is done by doing a background first and then the sample. It's a very low cost, and you can directly correlate that to the measurement that you are doing. For example, concentration of a liquid, a concentration of a matrix like a gas. For example, if you're a mixture of gases, you can directly correlate the concentration of a single component, and also used for solids and liquids. Then you have a total attenuated total reflection or ATR technique, which is now have become a standard. But there are certain pros and cons of it. One of that, it has to have a very good sample contact, and you can use various kind of crystals with it: germanium diode, diamond, zinc selenide, and that depends on the spectral length that you want, penetration depth, and robustness. You have specular reflection and also diffuse reflectance. And with a Raman, the first step to produce a Raman spectrum is to illuminate your sample with a light source such as a laser. This scattering of light is of a minute fraction of the light that you shine the laser with, perhaps 1 part in 10 million. And therefore you need excellent detector which can capture this low amount of light which has gained energy, for example. Most of the time, Raman scattering is designated as stocks where energy is gained. The Raman scattering is designated as anti-stocks. Most of the time, Raman light, as it is less intense than the stocks, anti-stocks are not used. However, it does represent equivalent vibrational information of the molecule. Here is an example of dextrose monohydrate spectrum acquired by all 3 techniques. As what one can observe here, near infrared spectrum shows very broad peaks. It has lower intensity compared to the Raman and FTIR. In order to analyze this kind of a spectrum, you would look at either the full shape of the spectrum, or you look at specific absorbance level on the X direction. Because the spectrum is so broad and peaks cannot be easily identified, in order to gather the information from such spectrums, you have to use certain algorithms. For example, PLS for quantitative. However, FTIR and Raman spectra, as you can see, are so specific. You can exactly pinpoint which peak belongs to which vibration. You don't need that extensive sample pre-processing and algorithms to use. Raman and FTIR spectroscopy differ in some key fundamental ways. So Raman spectroscopy depends on a change in polarizability of a molecule, whereas IR spectroscopy depends on a change in the dipole moment. Raman measures relative frequencies at which a sample scatters radiation, unlike IR spectroscopy, which measures absolute frequencies at which a sample absorbs radiation. FTIR is sensitive to heteronuclear functional group vibrations and polar bonds, especially OH stretching in water. Raman, on the other hand, is sensitive to homonuclear molecular bonds. For example, it can distinguish between C single bond C, C double bond C, C triple bond C bonds. Both methods, infrared and Raman, can be used with microscopic techniques. The key advantage of Raman spectroscopy is that it requires little to no sample preparation, while the FTIR method has constraints on sample thickness, uniformity, and dilution to avoid saturation. FTIR does still pose an advantage, which is in dealing with interference. Sometimes fluorescence may interfere with the ability of taking Raman spectra, which would not be an issue with an FTIR. Whether using Raman or FTIR, both have advantages and limitations, but when combined, these two methods can become a very powerful tool when performing materials characterization. There are a few takeaways that we can take from what I said before, and this slide really summarizes this. So FTIR, FTNIR, and Raman all in space, and when combined together with either of the techniques or two of the techniques, it gives a powerful tool for material characterization. FTIR really depends on the change in dipole moment. It does measure absolute frequencies, and it's very selective. As fundamental vibrations are observed, you can really pinpoint exactly which peak represents what molecular vibration. Although penetration depth is few microns, you can do most of the raw material ID with FTIR. Sample preparation and contact of the sample is needed, especially when using attenuated total reflection accessory. FTNIR can be used for bulk sample measurements because the near-infrared beam penetrates deeper into the sample, so you can measure the bulk sample as it is. No sample preparation is needed. You can measure through glass vials, blister packs, different kinds of polymer packaging like polyethylene, for example. Raman gives best of both worlds. Raman has excellent selectivity, and it does not require sample preparation. Of course, you can do the measurement in a non-contact way. Penetration depth is almost same as FTNIR and is complementary to IR. Only deciding factor is whether your material undergoes a change in polarizability. And also it measures relative frequencies at which a sample scatters radiation. Here in this example, we are showing our Summit FTIR, Nicolette IS5 FTIR spectrometer, FTNIR Antares 2, and this is DXR3 ThermoSmart Raman spectrometer. Now moving to vibrational spectroscopy implementation guidelines. Compendial testing is one of the requirements in various pharmacopoeias. These are standardized methods which are used for testing of raw materials or finished products. And testing and compliance to the standards are detailed within certain classic basic methods, which are related to the manufacturing, release, and distribution of pharmaceutical ingredients and drug products around the world. Various pharmacopoeias exist in the world. We all know about it. US pharmacopoeia, British pharmacopoeia, Japanese pharmacopoeia, Chinese pharmacopoeia, European pharmacopoeia. And they define a way to test an article or a final product. Here is such an example of what is involved when you have a diclofenac sodium, what you have to toast, which test you have to do, for example, identification test, and what sort of analytical instrument do you have to use. So for example, here, first identification AD, A is infrared absorption spectroscopy or spectrophotometry, and the article number is given as well. There are certain times where absolute identification cannot be achieved by a single test, and therefore you may have to do another test to really qualify that it is the article that you are measuring. And for that, they again can give a guideline on the secondary test that you need to conduct, for example, whether it's a visual test or another chemical test that you might have to do. As we understand, compendial tests are performed to determine if the material specifications are met or to address anticipated regulatory concerns. This test series can be also applied to determine general duct compatibility or for routine quality control. A validation of a compendial procedures. So USP 1225 chapter and EPICH Q2R1 guideline clearly defines the validation of the method. Validation of the method is to prove through a series of studies that a particular method will meet the intended analytical application. And those studies being that of accuracy, precision, specificity, detection limit, quantitation limit, linearity, range, ruggedness. If you have to implement vibrational spectroscopy tools for any of the compendial testing, then validation is a must. So for example, if we want to implement Raman spectroscopy for identification testing, then we have to determine first what category of test it is. From here, we can say it is a category 4 test, and for that, we have to validate the method for its specificity. Once you have demonstrated the specificity during the validation, it can be used as an alternative technique. If the method is of quantitative nature, where you are doing an assay, then of course a different category of the test applies, which is typically category 2. And then for that, you have to validate the method for its accuracy, precision, specificity, and others. You have to take into account when doing a validation, the analyst who is going to do the test, the instrument that you are going to carry out the test on, if you're using any reagents, and the matrix that you're going to use for the tests. USP also clearly defines the verification of the validation that you have already done. This is in chapter USP 1226, and also the transfer of these analytical methods are done by using the guideline in USP 1224. Typical industry practices for verification of compendial procedures is to look for specificity, purity, metrics. And when this verification is done, it's typically done to make sure that the method that you have validated still performs within the metrics of the sample that you have defined, with all the accuracy, precision, specification that you want, and which meets your primary analytical needs. A transfer of analytical method 1224 is typically required when one method is transferred from one lab to another lab or from one instrument to another instrument at a different site. And there are several things that you have to look out for. One is a comparative testing, so you have to make sure you have done the method testing on both the instruments, or you may have co-validated the method by taking the data from both the instruments. If that is not done, you have to revalidate the method on a second instrument, or you already have a transfer waiver because you already did such transfers in the past and you did provide all the information before. In order to do this validation and verification, there are certain statistical tools that are needed. I'm not going to go very deep into this, what are these statistical tools, but they are generally available on a USP website, and the chapter is USP 1210. And they basically focus on how to establish analytical performance characteristics for accuracy, precision, detection limit, and so on. They give you well-defined methods and criteria on how to do this test. Other analytical performance characteristics noted in USP General Chapter 1225 are out of scope for especially this chapter 1210. And USP 1210 also discussed the equivalency testing. So equivalency testing is done, for an example, when a pharmaceutical analytical test has been replaced by another technique. For example, a quantitative test for which you are doing a Kalfischer titration. Then the equivalency testing is to be carried out to make sure that the technique with which you are replacing the first primary method or analytical test is really equivalent to the primary technique test that you used to do with. Now moving on to the first case study, which is an identity test. So identity test in a pharmaceutical environment is carried out in different places. It could be either in a sampling booth or in a QC lab, for example. And mid-infrared systems, FTIR systems, are widely used for this analysis. Near infrared and Raman has its own space too. As you have understood from the previous slides before, near infrared and Raman has several advantages over FTIR, especially for non-contact, non-preparation of sample, measuring directly through the packaging. And therefore, many of the pharmaceutical companies are implementing all three techniques. They are mentioned in European Pharmacopoeia, US Pharmacopoeia. As you can see from the table here, various both pharmacopoeias have listed these techniques: mid-infrared, near-infrared, and Raman, and these are the general chapters there. And there are also the guidelines available on how to implement them. So what is main requirement for implementing a spectroscopy technique for identity tests? One is hardware and software compliance. So does the hardware have all the necessary requirements to achieve the identity testing as per the European Pharmacopoeia or US Pharmacopoeia? Does the hardware get tested according to the needs of pharmacopoeias? And is the software compliant? So is software 21 CFR Part 11 compliant? Is the data secure? Does it allow to implement e-signatures? And then can it be really easy to use for operators who are not spectroscopy scientists? So it should be easy to use. It should have workflows based on SOPs that can be created with the software. And can you easily configure reports and reporting to external LIMS, for example? So this is the main criteria on which the selection is done, of course. And here are our 4 typical units that are used for identity testing, which is a mid-infrared Summit FTIR, a near IR Antares 2, a standalone macro Raman, which can also have a probe for getting into the remote samples. And then we have IS50, which is our base FTIR, and it can be configured to all 3 techniques together. It can have a module for near-infrared spectroscopy with a probe. It can have a 1064 Raman in a sample compartment for doing the ID testing with Raman, and it can be also configured to have an ATR. So best of all three techniques can be used in a combined instrument in a lab, for example, or at a sampling booth to have a maximum coverage of your raw materials. Now showing the example of how easy it is to use such analyzers when you can automate the analysis with macros based on your workflow. We use several softwares. We use Paradigm software for our Summit FTIR. We use Omnic Spectra, or TQ Analyst, depending on the analyzer. For example, Omnic is a spectroscopy-based software, and it can be combined with Omnic Spectra to do the raw material identity analysis. In order to make this test as easy as possible, we can use Macros Basic to create a workflow. And that workflow could be, set up to collect a spectrum, collect a background first, collect a sample, so collect a spectrum, then use the library settings to do a search, like a QC compare or an identity test, and then save the data to a secure notebook, for example. So Macros Basic allows you to create your own workflow as shown here. And then we use something called MCR, which is a multi-component curve resolution software. Basically, it allows you to identify mixtures very easily if they are detected, for example. So here in the figure 2, we did an identification search without using MCR for a tablet, and we can see that we only found aspirin, for example. But when we did for the same tablet search with multi-component curve resolution algorithm, we clearly found the 3 main ingredients that were used in a tablet: aspirin, acetaminophen, and caffeine. There was a case study that we recently did for compendial raw material ID test. As we know, FTIR monographs for APIs are more prevalent for small molecules. However, they do not exist for NIR and Raman. The main reason being is FTIR is more prevalent and widely used. However, with the advance of Raman and NIR, these tests are more and more carried out using these 2 techniques. Here is an example whereby customer wanted to streamline their ID test. Cost of ID test was running into thousands. They implemented this during early product development and were able to file it as additional ID test, which is a type 1B for European pharmacopoeias. The utilization of Raman and NIR spectroscopy is typically filed as an additional, i.e. alternate to ID material testing and not as the primary method. Basically, this technicality avoids long filing processes. In the initial phase of the implementation, we did carry out a cost-benefit analysis for justification of the project, and it turned out that customer was able to get full return on investment in less than a year. And additionally, they achieved 100% inspection of their raw materials, and there was significant savings of man hours. Now we are moving to some in-process testing examples. For example, PAT QBD, that includes replacing Kull-Fischer titration, replacing gas chromatography for solvent exchange, and application of near-infrared in hot melt extrusion. One of the general concerns that relate to the Kull-Fischer titration is the generation of the hazardous halogenated waste during analysis. Thousands of samples are being tested daily worldwide using Kull-Fischer titration. And it does have detrimental effects on the environment and also on the personnel that operate the chemical analysis equipment. They have to handle halogenated reagents as well as the generated waste. And the halogenated waste produced by processes such as Kull-Fischer titration for water content determination can ultimately pose corrosion risks to equipment, health risks to people, and damage to the environment too. Given the chemical reaction on which the Kull-Fischer titration method relies on, both the volumetric Kull-Fischer titration procedure and colorimetric procedure represent sources of hazardous halogenated waste. There are several traditional Kull-Fischer titration assays that are done every day in pharmaceutical industry. As you can see from the example shown here, so many of SEN with MUOH, H2O in this mixture, H2O in THF, DCM, and so on, H2O in ETOH, all of these can be easily done with FTNIR. Typical concentrations, you can go to ppm levels, whether they are measured in ppm or weight by percentage, both can be easily done using FTNIR. This is a slide that was done for a grid chemistry and a spectroscopy webinar given by BMS recently. And it gives you a really good idea of the advantage. FTNIR analyzer can perform many of the assays traditionally done using titration. Even if you're looking at hydroxyl value, acid value, iodine number, bromine number, moisture, and more. It's very easy to use. Liquid samples are placed in disposable glass vial for measurements. There are no reagents needed, and assay can be performed in less than a minute, for example. The advantages of using FTNIR to replace titrations are speed, repeatability, ease of operation, and maintenance. Workflows can be created using Macros Basic or use result software for ease of use. Here we have an example spectra of sample containing methyl tetrahydrofuran, ethyl acetate, isopropyl acetate, and water. Here we are showing the method statistics of methyl tetrahydrofuran and ethyl acetate. RMSEP, which is root mean square error of prediction for both methods, is less than 0.05. Time and cost savings from alone one project in a year could contribute for the whole project cost. Also, it allows labs to be greener and more friendly towards environmental impact. In this example, with a one single spectrum, with no reagents and run the sample as it is, we can get 4 results of these 4 concentration measurements that were required for this method. There are many advantages of Raman spectroscopy, especially when you are doing an offline Raman spectroscopy comparing to chromatographic methods. This includes no sample preparation time for Raman compared to several minutes for GC or HPLC, reducing contamination risk, and a Raman spectrometer like this, which can also hold a carousel auto sampler accessory, you can really automate the measurements for many of your samples. There is no sample preparation needed. You can directly measure through the glass vials or bottles, as you can see on the screen here. If you want to use at-line or in-line analysis, there is an option of connecting a fiber optic probe too. And here is an example of so many of these solvent exchanges tests that are replaced by Raman, which can be traditionally done using GC. And many times multiple parameters can be easily measured. There are different units. Sometimes toluene, for example, Acetone in toluene is measured using molarity, and acetone in DCM could be measured in weight. But Raman spectroscopy can really meet the goals of the primary analysis for ensuring precise and accurate results. Here in this example, replacing gas chromatography for solvent exchange, this is acetone in water. This is in biopharmaceutical downstream processing. The cycle time can be reduced from few hours to few minutes. And then the other example is measuring DCM and acetone, and we get an excellent correlation of 0.999, which is almost equal to 1, and the same here, 0.999, and very small error of calibration. If we look at the comparison of the time between GC and Raman, Raman is very fast. There are less number of errors compared to the GC. There is hardly any sample preparation time. So of course you have a spare time for doing another project or running concurrently other analysis on a Raman during the time when GC is busy, for example. When fully implemented, testing cycle time reduces from 1 hour to 5 minutes for this test. Hot melt exclusion is very efficient, robust, and powerful solvent-free processing technology, which is now being exploited by many pharmaceutical companies because it promises the drug delivery that enhances the bioavailability of poorly soluble, crystalline, and potent active pharmaceutical ingredients. Crystalline APIs, they pose several challenges, including a poor dissolution, incomplete absorption, and reduced bioavailability. However, with the implementation of the hot melt extrusion, these challenges can be overcome. Extrude is versatile in its end use, and there are various controlled release delivery formulations that can be created. For example, immediate release tablets, controlled release tablets, orally dissolved tablets, soft gel capsules, thin films, stick packs, implants. It also allows to implement the continuous manufacturing. And for the continuous manufacturing, there are drivers, because you need to completely understand the critical process parameter. You need to have a full production control over its steady state, and you need to know various process variables that can affect the product. HME is really well suited for implementing continuous production and therefore QBD, quality by design, and PAT initiatives. The advantages of FTNIR in HME is FTNIR will provide chemical information of the sample, concentration, blending ratios, interactions between API and excipients. It's also sensitive to physical properties of the sample, such as color, temperature, and density, so you can effectively look at your process changes, and it provides incisive, responsive, and comprehensive process indication for your product quality control. So in this case study, we looked at a polymer-based API. The considerations were given to derive a partial least square model. What we did was we built a PLS model to monitor API concentration for HME. So for example, this figure shows the NIR spectra overlaid of ketoprofen, eudragate, and 50/50 extrudate. Both ketofen and eudragate are powders at room temperatures, and their absorption spectra were measured using an integration sphere of the Antares-2 spectrometer. The spectrum of the extrudate was acquired with NIR HME probe during the extrusion process. All spectra have strong features in the first overtone CH stretching region. And in addition, both U-drugate and the extrudate have noticeable peak features in the first overtone region. NIR spectra, as you can see, are quite often complex, and they have broad overlapping absorption bands, as you can see here. Chemical, physical, and structural properties of all species present in a sample typically influence the measured spectra. As a result, the NIR spectra depend on more than one variable simultaneously. Hence, a multivariate chemometric uses mathematical and statistical procedures such as PLS and principal component analysis for multivariate data. The spectral data was collected during steady states, including those at different temperatures, were used as calibration samples. NIR spectra were collected at 20-second interval, and 85 spectra with 7 concentration levels ranging from 40 to 60 were used for creating a calibration. There was a min-centered pre-processing done on the spectra, and the method created is demonstrated here, PLS model for API prediction. We got coefficients of 0.998 and root mean square error of calibration 0.43%, and that of prediction from the validation group is 0.62. Once this model was created, it was applied to the spectra collected during the HME process, and the results are shown here. As can be seen, the predicted API tracks the pre-blend API ratio with a feed rate of 100 grams per hour. As the process has a residence time of about 10 minutes, it takes 2 to 3 multiples of the residence time to establish a new steady state after the pre-blend ratio is introduced. This model was also subjected to a temperature variation change to make sure that model was robust enough to still predict the correct API concentration. And we came to know from this that even though the temperature change was plus or minus 10 degrees Celsius, the steady state temperature, we were accurately able to predict the API content. Process dynamics due to temperature change. This graph is very interesting because it shows how the NIR spectral response change when a different temperature setting is achieved. The sample spectra form a very tight cluster of principal component points. All these blue dots are principal component points in the PC domain. Once the process was stabilized, a 10 degrees Celsius temperature decrease was introduced in the temperature controller. The spectral response from the hot melts changed accordingly. As a result, the resulting PC data point moves away from the previous cluster. It took about 6 sampling times, or about 2 minutes, for the sample PC point to move from 120 degrees C cluster to a new location and start to form a new cluster. Since the PCA method relies solely on accumulation of multiple batches of good process spectral data to define the steady state, it does not require a calibration model. It is independent of the product formulation, and it's easy to implement. Finally, because NIR is non-destructive and it's so fast, NIR-based quality monitoring methodology can be easily transferred, from process development to manufacturing. Now moving on to finished product analysis, where we are going to see some examples of a PDMS assay with FTIR, replacing primary techniques like HPLC with Raman, and also look at the examples of contamination or foreign particle analysis and a distribution of a drug in a tablet with Raman microscopy. PDMS assay using FTIR involves an SOP. As you can see here, the steps that SOP has are quite lengthy. This covers sample preparation, analysis, and calculations. So this is a sample preparation, then how the analysis is to be done, and then how the calculations have to be done. This assay could be applied to all the pharma products containing simethicone. It's a lengthy procedure if really done manually. How can we reduce this procedure and make it efficient or streamline it for all the operators so you don't introduce any errors in your sample steps or calculations? One way is to create an automated workflow by using Macros Basic. So in order to do this assay, you would need Omnic, Macros Basic software, Omnic Spectra or TQ Analyst, depending on what the calculations you are doing and which method you are applying, and an FTIR, of course. With the Macros Basic, one can create a workflow. This workflow will guide operator on each step of what to do. So for example, this workflow starts with a PDMS assay, clears the screen, it asks for the information for a sample, does some initial math on it, and then it collects the spectrum, it displays it, calculates the peak heights, does few more calculations, and then it stores the result where it should be, and then the report printed or saved. And here is an example from such automated workflow. It reduces the time significantly, it reduces the errors significantly, and all the operators will have the same feel of the workflow. And one can train an operator very easily, less than a 2 minute, on how to run this macro. So streamline and maximize the efficiency with Macros Basic automated workflow is demonstrated by this example. If you have any other analytical challenge, one can easily accomplish that with the Macros Basic, whether you use FTIR, near-infrared, or Raman instruments. Another case study is to look at the replacement of HPLC assay for 2 APIs within Raman spectroscopy. We were given a task to look at application study which involved measurement of 2 APIs for a single oral drug product. For these 2 APIs, one HPLC assay was used, and the customer decided to improve some of the analytical processes, and this product and this test was chosen because it was first easier to implement and they could replace completely one test using Raman spectroscopy. Idea was to implement this at multiple sites, recognize the cost savings, and gain and have maximum gain in terms of the ease of implementation, time, and of course the cost savings were also considered to review the new analytical method. For that, DXR3 Smart Raman spectrometer was used. As you can see here, Smart Raman spectrometer has an automation capability. You can run several tablets using a tablet holder and automate the measurements. You can do this measurement in a 98 well plate for high throughput analysis, or if you have vials, you have a carousel rotator and you can automate the measurements for all different types of samples in this kind of vials. If you want to do a standalone measurement for a one single glass bottle or a one single glass vial, it can also accommodate that. DXR3 Smart Raman system can be used along with the Omnic XI software, TQ Analyst for quantitative method or qualitative method, and can be totally automated by using Macros Basic. So in the case study here, we are showing a spectra of 2 APIs, API A and B. This is the calibration spectra of oral drug. And then these were the validation spectra of the oral drug. Customer did supply a separate set of calibrations data and validation data. So separate set of calibration samples and validation samples with the reference analysis. When we look at the results, PLS model API A, for API A, we created a partial least square model, and we got a root mean square error of prediction 1.40 with correlation coefficient of 0.996. And for the API B, 0.9929 was the correlation coefficient, and root mean square error of prediction 1.91. Models were built with 7 factors for API A and 4 factors for API B. When we look at the validation results, we can clearly see the actual API and the API calculated, and the API B actual and API B calculated. Typically, if you want to compare the two data sets for probabilistic testing, an easy test is to see whether these two data are in agreement to each other. And for that, we do an F-test. And when we do an F-test of API A, we got 0.949, very close to 0.95, and F-test API B, 0.99. So we can clearly say that the Raman results that were given by this PLS method are actually correlating to the actual reference values, and therefore this test can be a really good candidate to replace the HPLC test. When looking at the return on investment, for such application, the return on investment is very quick, because the yearly expense of the HPLC solvents and the waste disposal of solvents can be so high that if a purchasing manager calculates those two costs together, that will cover the whole project cost. But to be conservative, keeping in mind other things involved as well with the Raman, like maintenance and some application time, still we can get the return on investment in less than a year, and here in this case it seems to be 8 months. Next, we have to look at the case study of API distribution in a tablet. Many issues from the field of pharmaceutical and medical technology research and development require the use of surface analytical techniques. For example, looking at the API and excipient distribution, residues, contamination in ampoules, particles, discoloration of blister packaging, residues from cleaning agents, in syringes and needles, distribution of active pharmaceutical ingredients on stents, for example, implants or patches. The drug form itself can also be the subject of surface analysis. Here we want to show the example of tablet imaging for analysis of the distribution of API in a tablet. It offers the possibility of identifying different molecules of API and excipients using Raman microscopy. Moreover, the tablet can also be measured confocally to look at the depth profile of the API or excipients. With Raman imaging, the dosage form and the API distribution can be shown in detail in the distribution image. Multivariate curve resolution, or MCR, with constrained alternating least squares is a powerful method for deconvolving overlapping spectral signals from chemical and biological reaction systems. The intended purpose is commonly the estimation of concentrations of individual components or the identification of unknown spectral profiles in complex mixtures or aqueous solutions. Generally, MCR has the ability to estimate both simultaneously from a spectrum. The specific feature of MCR is the decomposition of a spectrum in a physically or a chemically meaningful way. With the power of MCR, you don't need high quality single spectra. They can be very quickly calculated from a spectra taken from a Raman spectroscopy, a few seconds, and from the data set we can then calculate the component percentage area. For example, this is a spectrum in the blue taken very quickly of the sample, and then the area calculated live, while you were watching the sample. So for example, here 33.87% was the area coverage of a D-mannitol on a sample surface. When we do an MCR, if we do see both peaks of A and B, for example, here in this case, ibuprofen and mannitol, I assume that every spectrum in the tablet is a linear combination of the spectra. Here in this case, from mannitol and ibuprofen. When we calculate the correlation between the calculated linear combination and the actual spectrum, we look at the overall area and the mix and the composite spectrum of ibuprofen and mannitol. Once we have completed that, we can get the sum average of the respective coefficients to obtain the total percentage coverage of ibuprofen and mannitol. This approach makes this analytical test again very quick. If you had to create a PLS equation for separately looking at ibuprofen and mannitol reference, one had to create a calibration based on PLS with several lots of tablets. But with this approach and automating the MCR analysis, this result can be completed in less than an hour. So here in this example, we ran the MCR on a tablet. We had 100 micron particle size. Filler particle size was 211 micron, and the API concentration was expected to be 20%. We ran this experiment for 29 minutes with 10 milliwatt laser power, and we just did one scan, basically at each point. We achieved 462,000 spectra with a step size of 15 micron, and we used a 10x objective, and the frequency of the collection was 300 Hertz. Here in this plot, figure one shows the correlation between the calculated linear combination and the actual spectrum. Since we collected a square area and the tablet was round, it was clear that correlation was lower in the corners of the tablet. And it showed a small defect on the tablet itself. So we excluded the spectra below a certain threshold from the concentration calculations, so that spectra in the corners like this are not taken into account when calculating the percentage API and the percentage excipient. The second and the third plot are in essence of the concentrations correlations of ibuprofen reference and mannitol reference. And the percentage displayed are some normalized concentrations based on the weight coefficients. As you can see here, API and excipient distribution with a semi-quantitative result. When we say semi-quantitative, it is coming from the weight percentage of the actual API and excipient in terms of the spectral weight percentage. And therefore, you don't need a very cumbersome PLS method. You can almost see the correlation between actual API concentration and what we have measured. So this is a very good way of clearly checking the quality of the manufactured tablet without spending so much time on method validation and calibration. But it does give you a guarantee that your API is distributed well. It has the required percentage of the API, and there are no defects in the tablet. This result we got in less than 30 minutes. Now moving to detection and identification of foreign particulates in liquid formulations, parenterals, or injectables. There are certain distinctions that must be made between intrinsic and extrinsic particles in the manufacturing. Intrinsic particles could originate from the medicinal product or API itself. They are undesirable, but may be part of the medicinal product and described in authorization. However, extrinsic particles or foreign particles originating from outside, such as fibers, glass, abrasion of machine parts, are undesired under any circumstances. During the testing process, if foreign particles are found, they have to be identified because, first of all, it initiates a non-conformance or OOS observation, and that batch goes on a hold, and therefore it is very apparent that such a particle is identified and the root cause of analysis is initiated to mitigate the risk of repetition in the future. For this, several techniques can be used. Infrared microscopy, here ion 10, Raman microscopy, DXR-XI or DXR, Raman microscope, and a complementary tool, SEM-EDX, can be used too. SEM-EDS is a complementary technique to Raman or IR, which can classify particles, mostly metallic or inorganic, size 1 micron and greater, and it provides elemental composition of each particle. SEM-EDS can identify additives or impurities present at trace quantities that may not be IR active or are present in insufficient quantities to give IR information. The combination of Raman and SEM-EDS allows a comprehensive understanding of the many different types in a sample population as a whole. This includes organic, inorganics, and metallic particles. EDS with FTIR allows us subtle differences in trace elements present in test articles that are made of similar chemicals, for example. We also have a standard FTIR or a small smart FTIR that can measure particles from 5 mm to 100 microns. Particles from 100 to up to 10 microns can be analyzed using FTIR point and shoot microscope or imaging system. And particles from 100 micron to 1 microns can be analyzed using the XR3 smart Raman microscope. This is a slide which is showing an example where we were given parenteral samples with foreign fibers and particles. We had to identify all the foreign objects and also a residue of dark liquid in the sample. We did this with ion 10 infrared microscope. The ion 10 allows users of all skill levels to easily and confidently perform their microanalysis function. With the microscope, Omnic Picta user interface was used, and users with little prior experience in microscopy or spectroscopy are able to quickly and effectively collect sample data to characterize a microscopic material. Here in this example, first a spectrum of a filter was taken, then the fiber was taken, and a black spot was taken. Taking the spectrum of the filter is to subtract the filter from the composite spectrum. So for example, when we analyze a black spot here, we get a composite spectrum of a filter and a black spot itself. And then when we subtract that, we can see actually what this black spot is made of. And when we did that, we can see that it was some oil related black spot. And then in the next slide, we can show you actually what the fibers were. So spectra contaminant function is ideal to do this type of analysis, whether it's a foreign object or an intrinsic particle arising because of the process difference or the process initiated change. For example, in biotherapeutics, it can happen. So a black particle on the filter, this is done with ATR, and the picture in the red shows you, highlighted in the red shows you the area where we are taking a spectrum. This is a real image or a photographic image of the area that we are measuring, and we can clearly see that at that position we had a microplastic, I believe polypropylene, and then we can look at the correlation map and identify all the particles here, and we can ascertain at which position if there was any other fiber present, for example. There are two modes of operations for particle analysis. One is a manual selection of particles, or automated with reference to the position and size of the particles. The particle wizard automatically allows the analysis of the region. After the region, the video image is selected, the software identifies the target particles and proceeds to produce spectra for each particle. So this is a typical process. Here in this slide, we demonstrate two different modes of operation: analysis with an ATR and analysis of the same sample in transmission mode. When a sample is analyzed using an ATR mode and compared to the libraries, we can clearly get a match for PBT, polybutyl terephthalates. And when we switch the mode, because the mode can be easily switched from ATR to transmission, and transmission mode can be used when you can really get the light transmitted through your sample and it's quite thin. And in that case, we can see one more component which was present, which we did not see with an ATR, and that was an adhesive that was used, for gluing probably two polymers together as a laminate, for example. So this came as a thermoplastic elastomer glue when we looked at the particle in transmission. This way of utilizing both the techniques and switch of the mode very quickly from ATR to transmission gives you a very comprehensive identification of particles, which you cannot get if you use a single ATR technique. We have a global service and support. We have over 2,000 service specialists globally, and our average experience of field service expertise is 18 years. This is unmatched in the industry. We provide wide variety of services, after sales support contracts, application assistance, remote digital support, diagnosis help, application support at customer labs or our labs within various locations, and unmatched support in terms of the product need that you have or feasibility studies that you want to carry out. In the phases where we don't know an application, we can help you to do the feasibility test either in our lab or a customer site. We typically loan the instruments out for this kind of test, but where the instrument sizes are bigger, we carry this out in our lab. So for unmatched service support, feasibility test before sales and after sales, please feel free to contact us. So with that, I thank you, and we will start a quick poll, and after that, I'll take your questions. Thank you.
Thank you, Shailesh, for your excellent presentation. Before we move on to our question and answer session today, where we will try and answer a few questions, we have 3 poll questions to ask you. So, number 1, which of the molecular spectroscopy techniques do you use for incoming raw materials testing? FTIR, NIR, Raman, USP wet chemistry monograph testing, or all of the above? Please vote now. Thank you for voting. It looks like the majority of you are currently using Raman or FTIR. Our next question for you is, what is your primary go-to method for following a chemical process? HPLC, LC and MS, NMR, inline Raman, NIR, FTIR, lab Raman, or NIR? Thank you for your participation. The most popular primary method for following a chemical process seems to be HPLC. And our final question before we do move on to our question and answer session is, which chemical imaging technique do you use to verify uniform distribution of drug in solid dosage forms? IR microscopy, Raman microscopy, SEM, SEM-EDS, or none of the above? Thank you once again. It seems like the majority of you are using techniques alternative to the ones described in this poll. So let's move on to our question and answer session, where today's speaker, Shailesh, will answer a few of your questions before we have to finish. Don't forget, you can still submit your questions using the questions panel in the menu on the right. So let's take a look at our first question. Shailesh, can you tell me, can you use Raman or FTIR to determine the solvent contents in drugs to the permitted levels?
Thank you for the question. The question can be really answered as long as you can, prove within the calibration and validation studies, first of all, the specificity that the solvent that you want to measure, you can see within a near infrared, Raman or infrared spectrum. And the second criteria would be, of course, whether you can get to the levels desired as per the HPLC or coefficient titration that you want to use. So of course you can. And the second question related to the same person, I believe, was whether FTIR or Raman could be used for such analysis. And yes, of course, there is a sampling need for FTIR, so you need to, have a transmission cell and then sandwich the liquid in between, and you can do that as well. You can get to the ppm levels, in some cases even ppb levels. And with the Raman, because it has a confocal aspect, you can measure through thick glass bottle, for example, directly. Again, the validation does play a part here, so you need to make sure that it's accurate, reproducible, linear, and robust to replace the primary technique.
Thank you, Shailesh. And one more. How would specificity be demonstrated using Raman methods instead of HPLC methods? Could you elaborate a little bit on this?
So A USP 1225 validation criteria clearly describes when you use an alternative technique how to determine the specificity or a selectivity. When you're doing a raw material ID and your primary method is HPLC, you have to make sure that the raw material that you measure does not give a false positive for other very likely candidates. So for example, we are measuring lactose and a customer also has a lactose a monohydrate and a dihydrate, then you have to make sure that dihydrate does not pass as a lactose monohydrate. So a selectivity study done during the validation for a ID requires only specificity to be tested, and if it's specific, then you can replace that. Specificity for quantitative type of tests, which are quality two tests, involves creation of assay and a placebo, and make sure that Raman does not give an answer for something that is not there. So doing a principal component analysis first, and then determining that the sample that you are measuring is not outside the scope of the calibration can give you that additional security.
Great, thank you, Shailesh. Unfortunately, that's all we have time to discuss today. Any questions that we were unable to answer due to time will be addressed and followed up later via email. Please join me in thanking Shailesh for his time. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please, can you take a moment to provide your feedback? If now is not a good time, the survey will be sent to you shortly via email, and we would very much appreciate it if you could complete it when you can. On behalf of European Pharmaceutical Review and Thermo Fisher Scientific, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.